Hello, my name is Daniel Tsai, and today I'm presenting my project titled Investigating D. melanogaster cell sheet morphogenesis. I've been working on this project for over 2 years now as a member of the Keyhart Laboratory. Dorsal closure is a 3 to 4 hour process midway through embryogenesis in D. melanogaster involving 2 sheets of lateral epidermal cells zipping an elliptical hole filled with amnioserosa cells on both ends along the dorsal midline of the embryo. I use DC as a way to illustrate cell sheet morphogenesis in D. melanogaster, which shows many processes at the molecular, cellular, and tissue level with humans. As of 2018, there were over 140 known dorsal closure genes, which are defined as any gene that causes an abnormal DC phenotype when mutated. The goal of this project was to use a Bloomington D. melanogaster deficiency kit to identify DC genes in 2 of the 49 deficiencies of a recent 2L screen that produced significant DC defects when homozygous in a UPK-GFP background. To be clear, a deficiency is a mutation in D. melanogaster that removes long stretches of continuous DNA on a chromosome. I dissect each deficiency by performing multiple trans-heterozygous crosses with partially overlapping sub-deficiencies as noted in the crossing scheme below. Trans-heterozygous embryos undergo a time collection, then they are manually sorted and mounted on an inverted confocal microscope. Over 120 time-lapse images are captured over a 4-hour period for each embryo. The first parent deficiency, DF27, underwent a cross with DF27A. This cross partially phenocopied 2 of the 3 major defects of the homozygous parents. Therefore, we can assume the genes responsible for those defects, which are cuboidal LE cells and a cigar-shaped dorsal opening, are located within the 54 kilobase pair 8-gene homozygous deficient region of the cross. 2 genes I hypothesized are BOL and BARC. Correspondingly, genes responsible for the ASL falling apart defect and overall severity are located within the 64 kilobase pair 11-gene heterozygous deficient region. The second parent deficiency, DF38, underwent 4 crosses with DF38A, B, C, and D. The cross with 38A partially phenocopied the jagged leading edge defect, 38B partially phenocopied the abnormal AS cell-shaped defect, 38C partially phenocopied both the jagged leading edge and abnormal AS cell-shaped defect. Lastly, 38D partially phenocopied the abnormal AS cell-shaped defect. 3 major conclusions can be drawn from these results. First, genes for the abnormal AS cell-shaped defect are located in the 8-gene overlap between 38 and 38B, C, D. 1 candidate gene I've hypothesized for this region is LAM. Second, genes for the jagged leading edge defect are located in the 2-gene overlap between DF38 and 38A, and 4-gene overlap between 38 and 38C, but not B or D. 2 candidate genes I've hypothesized for this region are ocelin and CG14024. Lastly, severity in the parent deficiency is a result of a combination of multiple genes located throughout 38. 3 candidate genes I've hypothesized for this are fusyl, CYP, and MID. Moving forward, alternative crosses with mutant alleles and rescue constructs will be used to study the role of individual candidate DC genes. I want to thank everyone in the Keyhart Laboratory and, of course, all the flies who dedicated their lives to this project. And with that, I thank you for listening. I hope you enjoyed learning about fruit flies as much as I have.